So we're collecting these sediment cores because a couple of groups are interested in the microplastics and analyzing the different layers of sediment on the seafloor and if the microplastics might change with those sediment layers, as well as correlating them with the microbial communities that live in there that could potentially break down microplastics, but that's what the research is looking into to see if that changes with depth within the sediment core. And then one of our partners is the U.S. Geological Survey, and they're interested in measuring particulate organic matter, or POM for short, because they're also interested in piecing together a food web. So who are the players within this food web? And this would be the base for the detritivores, and that would inform the other hierarchical layers of the food web. If we understand what's in the sediment, we can trace those stable isotopes up through the food web from the detritivores and up from there. So we can source where different parts of the food web are getting their food from, essentially.
And Rennie, just so you know, these are actually going to be labeled as two different samples. So these 10 and 11.
Okay,
yeah.
Sure. So 10 and 11.
Get a zoom in just a bit more, get a screen grab. There you go.
Great, thanks.
Got it?
Yep.
Looks great.
Those jaws look open ridiculous, like a little bird waiting to get fed.
Yeah, they open pretty darn wide.
They do. It's crazy. Wow, stuck in there pretty good.
The USGS is also subsampling a lot of the corals and sponges we've been collecting as well to understand their stable isotope signatures, to piece together this food web. So this is just one piece of a much larger puzzle with the studies they're doing to better understand the deep sea and how it might be connected to shallower parts of the ocean.
That was a great explanation of why we take these push core samples. Lizzie, thank you.
Yeah, of course.
Read my mind, Jake.
You like the Argus zoom? It's a good shot.
So this is the Bob Waters method?
Well, you could see the giant dust storm that I really had a hard time getting that first one out.
You did a good job of pointing into the wind like you wanted.
And it was a big giant dust cloud when it came out. As Steve says, we're pointed into the wind, so. But if the other one wasn't in, that could have potentially contaminated the site, obviously. I don't know if it's only Bob's method or OET or.
Yeah, I just
trademarked.
Yeah, kind of heard it trickle through the pilots. I think its origin was Bob Waters.
But that's cool.
Yeah, that makes a lot of sense when you think about it.
Zoom in just a bit for us, Steve.
Especially if you're interested in the surface.
Screen.
So at home, what they're talking about is when they lift up a sediment core and you kind of get this plume of the finer, more friable, soft sediments that may come and land on the site you're interested in, this would be really important if you're really focused on that very top layer and comparing that to layers below it, because as you saw, as Dan brought up that other core, it kind of sprinkled deeper layers of that sediment over the surface, so it could confuse or mix up your signatures if you were doing very precise measurements with layering of the sediment in this core.
And that's what the scientists typically do. They do very fine scale analysis.
This one might stay.
It's trying to pull it back out as I come out, so I was trying to wiggle it around and kind of break off the core there, if you will. But I don't know if it's going to stay in there. Seeing a little puck fall out there.
Yeah, is that still enough for you then?
Oh, it fell out.
Yeah, that'll be enough for me, and then the other group can have the bigger core.
Oh, it's still in there.
Okay, sure.
Yep.
I thought it fell out.
Darker top layer. Can't really see it.
Yeah.
Yeah, I was just about to say, you can actually see the difference in the coloring.
That's a piece that fell out. Sorry.
Just a small puck.
Like a rock.
Yeah. That's why you had a hard
Looks
time
like,
pushing it down and bringing it back up.
A
Sausage
hockey
patty.
puck.
It's like something I feed my dog for breakfast.
You don't feed your dog kibble?
No.
Oh, he gets something out of a can, I guess.
No, they get raw food.
Nice.
Wow.
They're spoiled dogs. We make up a special concoction, and it's kind of like a sausage thing, and we freeze it up. Let's see if we're gonna pick it up. It's really hard.
What about the other hockey pucks?
It's like that scene in Edward Scissorhands.
Okay.
All right.
Porch is still out.
What?
Or not porch, sorry. The starboard bin.
The box.
Okay, back continuing eastward. That'll be
Warp
directly
speed.
upslope.
Yes. Make it so, number one.
Okay, so there was a bit of layback, so we can keep going at 0.5, but it's a bit, it takes a little bit to stop. Bridge nav.
Maybe we should do 0.3 now. It's looking pretty steep there.
Yeah.
At least to start.
I concur.
Yeah. And if you have the sonar on and you can sort of see.
100 meters east, speed 0.3.
You also have a full wrap right now. I don't know if you want to.
Really?
Yeah.
Is that me or
That's you. Yep.
Pinhead? Okay.
So
So I need to go this way.
left. Yeah.
Thank you. How did I do that? I always tend to turn the same way. Just in case you wanted to look at that crab back there.
Pretty big.
And I did.
Ooh.
Thank you.
The size of that thing.
What? Are they tasty? Do we eat these?
I don't know. I could pick them up and stuff them in the bio box and find out.
Post-shift snack.
I think that this, I remember hearing them narrate, this is a type of neo crab. I'm going to look it up. I can hear myself echoing somehow.
We don't hear an echo on
I
our end.
can't. Yeah.
Yeah, do you have a window open that's not muted?
Hmm.
If you opened up a different window to see satellite 3 or
Okay,
something.
headed east.
Thanks, I'll check.
Kurt is saying Neolithodes. Lithodes? Scarlet king crab, he thinks.
Wow, it's up.
Yeah.
That's great. I think that's what we wanted.
Thanks.
I had also heard. Are you good, Lisa?
Yeah, I'm good.
Testing. A goblet sculpin or a blob sculpin
Blob
that
sculpin?
people might have seen. Yeah, that people had seen earlier.
Yeah.
That sounded really
Can
cool.
you reset one?
Yep.
Preset one, thanks.
Did you only see one or did you see multiple?
Of the blob sculpin?
Mm-hmm.
Oh, I didn't catch that. I didn't see one at all. I just was.
Oh, that's right. We're on the same shift. Ah,
Yeah.
that makes sense.
Yeah, they saw a couple of blob sculpins.
Those the little predator sponge guys there on each side of the.
Those, I think, might be tunicates.
Tunicate.
I'm not
Yeah,
sure if they're
that's-
sponges. They could be.
Zoom
They
in real
could be,
quick,
though.
Steve.
Yeah, I don't know. Tunicate or a sponge? Not sure.
Kind of looks tunicate to me.
Yeah, I just didn't.
As a,
Do they make those stalks like that, or is that an existing stalk that they're taking
that's
advantage of?
an excellent question.
Zoom in a bit more if you want.
Oh yeah, you can
Good,
kind of
good,
see
good.
through it. You can see its stomach.
Yeah.
And that's
Yeah, so Kurt is saying it's a stock tunicate.
Stalk tunicate. There we go.
it's like a little flag.
Deep
Awesome.
sea flower.
Thank you.
That's what
Yeah.
it looks like.
Mm-hmm. Little tulip.
Meant to be going east, right?
Roger that.
I'm pointed east right now. So.
Roger. Me too. A bunch of those guys.
They're like little weather vanes.
Yeah.
I have a question for the crew on the Nautilus right now. I'm really curious because of COVID and everything. You guys had some pretty strict quarantining protocols to make sure everyone was safe on board. How did you guys pass the time for those 2 weeks?
I was in San Pedro for it. We were in a hotel.
Were you mostly
not
in
far from the
hotels?
ship. Yeah.
Yeah. Okay.
So just went on a lot of runs.
Yeah.
Yeah.
I was in Seattle and we had a few days of very nice weather. And then, so I was going running and walking, and then the wildfire smoke rolled in and it was pretty brutal, and we could barely even go outside for like a week and a half. It was,
Yeah.
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 And does that mean all your food was delivered?
Yeah, meals delivered, grocery runs. It wasn't a terrible experience. I had some work to finish up from the summer, so I had a lot of time for that.
Yep. Luckily, the hotel I was in had a little kitchenette, so
Oh, nice.
could make.
Yeah, that was a nice hotel.
Yeah.
Yeah, it was a pleasant experience. It was fine. Interesting because I'm
different.
used to living with my housemates, so I see them like every day. So it was interesting to not really hang out with people, but I had a lot of work to do and would just FaceTime people or go for walks. It was a very beautiful area in Oregon where we were at. It was very nice.
And then it's a big shock to come back on the ship.
Yeah, I know.
And be able to hang out with people. You know, we're all wearing masks and everything, but still just
Yeah.
hang out with a lot of people. And then I know it'll be a shock going back to shore,
Yeah.
going back to
Yeah.
kind of reducing the amount of people you have contact with regularly.
Yeah,
So
those are
will
pretty
you have
extreme
to-
opposites.
And will you have to self-quarantine when you get off the ship, or?
I don't think it's not as mandatory as when we were coming on to not contaminate, bring on anything on the ship, but it is recommended that we isolate for a bit. I personally, with all the safety protocols and everything that's been going on, and the great job that OET's set up for that, I think it's much less risk that we would be on the ship and something would have happened. Really, it's more of a risk getting off at this point.
Yeah, I feel safer here.
Scary getting off.
I know returning to campus, I either need to quarantine or get tested.
Oh yeah, because
So-
you have you're
Yep.
in an institution.
Yeah, for anyone who's familiar with UC Santa Barbara, we just got notification that Isla Vista has 2 separate COVID spreader events kind of going on right now. A fraternity, a frat house, and a, what's the other one? Fraternity and,
Sorority?
Sorority.
there you go. Clearly I was never a part of one. A fraternity and a sorority house.
From what I understand, Santa Barbara was largely spared in the earlier days of the pandemic. It kind of hadn't really been spreading around up there, but that was a while back.
No, not as much. And it is concentrated in places like Isla Vista,
These darn
and then
students.
Santa Barbara County.
Yeah.
I know. Yeah, they're posting on the news. You just see pictures because everyone's just roaming the streets and hanging out together outside.
Youths.
I know. Those young people. Is that another Pharea sponge that we're seeing? I think so. Maybe a smaller one.
Yeah.
This is promising as we're heading upslope. Yeah, I've got a couple of different. Right on, right on. Hopefully we'll see more.
Yep. Good eye. I love seeing the shadows of the fish before you see them.
Yeah, if they're swimming in front of the lights, they cast an ominous shadow.
Yeah. And especially, with Halloween coming, it's very ominous.
Mm-hmm. Looks like
Right up there
another
with that
dead
dead
Pharea.
sponge.
Yep.
Quick zoom there.
Looks
Steve.
like another Pharea.
Yep.
Yeah, good, thanks.
Pharea with a crab
A lot
of
of
some
habitat.
kind. Very prickly.
Yeah. And same crinoids.
Okay, thanks.
Thank you. Thank you.
I'm gonna grab Bubble Dan for gauge checks.
Yeah, go ahead. Thank you.
There's the stock tunicate, I guess.
Yep.
More of the same.
I've seen quite a few of those guys now.
Yeah, and a few more asbestopluma carnivorous sponges. So hopefully we're getting into some more stable habitat where things can grow, and we'll see more and more of what we're looking for. Look at this, another dead sponge. This was promising on an earlier dive. As you're headed upslope, if you're seeing a lot of dead sponges, it's usually, but not always, a good sign that there might be more living sponges ahead, up closer to the top of the slope.
So I had another 100-meter east step, and then after that, I think to maintain going upslope, we'll have to alter course a bit to 115 or 120.
Yeah. So east northeast, we're hearing from Guy Cochran.
Oh, you want to
He's
go over
recommending, or
northeast
no,
then?
I
Okay.
think,
no. Wait, he said east southeast.
East southeast, all right, yeah. So we'll
Yeah.
continue just one more step east, and then we'll start to go southeast, up, continuing up this, kind of trying to stay perpendicular of the contours.
Yes. Perfect. Thank you.
Yep.
yeah, we are on an upslope mission.
Because I think we've seen a lot of this habitat already from the beginning of the dive, which started at, I think we hit the bottom at 7 p.m. So maybe we could just, without sampling for a while or doing transects, we can get to that other part, hopefully, of this structure.
Do you,
Yeah, I think taking,
so I'm sorry, did I hear that you do not want to do a transect? Is that correct?
No,
Okay, just checking.
I'll make that executive decision.
Sure.
Especially since the dive will have to be back on deck by 8 o'clock this morning.
Mhm.
So I'll sacrifice some transects right now.
We're kind of going at
Oh, and we talked
transect
about this.
speed, so if you ever want to throw one in there, we could always do it. But
Say that again?
we're already going at transect speed, so if you ever want to call one, we can just kind of continue on. But of course, it would reduce our ability to stop if you want to, without ending the transect. So
I think that's okay. I don't think we're interested in stopping much.
Yeah, that's fine. We can do a transect then if we're going at the transect speed. And we just won't stop and sample in
okay,
between.
yeah, sounds good. We can do that. It's up to you if you want to call it one or not. So we are continuing on this east. I will alter course and we'll just continue on transect, and it'll slightly alter the course of the transect, but that should be fine, I think.
Thanks for the suggestion. So just let us know when we'll start.
I think we're good to start here.
Okay.
Call that there. What number transect is it?
Transect 4.
We made it an hour and 45 minutes into the watch, Jake, without doing a transect.
Is that a record?
I think so.
For the
least
last
this leg,
few,
yeah.
last few legs, sure.
Yeah, well, and
On
Lisa
day-
and I had talked before this. It's really, this is definitely more exploratory mode, so more
Okay.
so than maybe some of our previous dives, just because we haven't been here yet. So we're, and I think,
we don't
let me
have
know
to
if this
do
is
it.
still true.
We can, if you want to,
I mean,
if you
if
prefer
we're-
to zigzag, then we could definitely just hold off on it.
No, let's take a transect right now, and then after that, if we're just going at the speed anyways,
Yeah.
and then we can zigzag.
Okay.
Yeah,
Thank
I
you.
think your suggestion was right. If we're already going transect speed, we might as well take one while we're going, because I think we are trying to go put some ground underneath us to get upwards. So zigzagging might take a little bit longer.
Yeah.
Two birds, one stone, might as well. But once we get to where we're going, if we feel like exploring more than doing transects, I think the reasoning behind that is just because we have not explored the deeper part of Santa Lucia Bank at all, and so we're fine with making that more of a priority than the transects, to just give you an idea of where we're coming from.
Sure. Sounds good.
Transect mode enabled. I think we cover more ground this way anyways, like the tortoise and the hare, right?
Yeah. Steady speed.
I have to push that blue button over there.
Yep.
You got a second?
So if anyone's just joining us, we are roughly 1,400 meters below the surface, and we're exploring depth near the Santa Lucia Bank, which is actually just northwest of Point Conception near the Southern California. Ooh, octopus.
Octopus there.
Yes.
On transit.
I should have never
Got it.
suggested it.
Yeah.
Yeah, I can blame me for that one.
We're counting you.
And so we're exploring the deeper parts of this bank and escarpment area. We're hoping to do several dives here to get a better idea of what kind of deep sea resources exist on the seafloor. In this area near the surface, it's extremely productive. There are a lot of whales in this area and mammals, and it's a hot spot for commercial fishing. But we don't really have a great idea of what's going on deeper towards the bottom. And so this is what we're doing. We're putting eyes on the seafloor, and we have new mapping data that's helping guide these dives safely. And we're exploring and kind of hoping to see some deep sea corals and sponges at the top of this ridge.
Yes, and all of this rubble, it had to come from somewhere. So Guy Cochran, who has been very helpful to us, particularly at National Marine Sanctuaries, he's been our geology advisor, basically. And he works for USGS, and he's saying that this rubble had to come downslope from some high-standing bedrock. So hopefully we will find that bedrock, the holy grail of this dive. And that's why we're just trying to move quickly to a location where we hope that bedrock is located.
Yeah, and one of the reasons we think that we'll find more deep sea corals and sponges on more consolidated bedrock is this rubble being smaller chunks of that bedrock are more easily disturbed or movable if there were to be strong currents or waves. Oh my goodness. Oh, is that a big
grenadier? Yeah. Or is it a flat? No, it's a grenadier.
Oh, it's humongous.
It's huge.
Yeah, the shape of his head was different. That was amazing. So as I was saying, we do expect to see deep sea corals and sponges on consolidated bedrock that doesn't move or is less easily disturbed, because deep sea corals and sponges are very slow growing.
I'm going to change course here to 120.
Roger.
Bridge nav. 100 meters, bearing 120, speed 0.3. Thank you. I think that ought to keep us a little more towards our upslope goal.
Yes.
They don't have multiple modes of getting energy and nutrition. They rely strictly on filter feeding. And so they don't require light to grow because they don't have that photosynthetic algae within their tissues. But they can grow extremely large. The deep sea is a fairly consistent environment. It's cold, it's dark, it's deep. And so they do require a fairly consistent environment to grow and be happy.
Yeah, I was sailing with Chuck
these
Fisher.
Smaller pieces of bedrock or this rubble are more easily movable by currents or maybe slope failures or falls from above, they're more likely to get turned over or sedimented, which would clog and kill filter feeders like corals and sponges. Although you will see some, like on the left hand, we have a red mushroom coral there, and they tend to be more quick growing and they're pretty darn ubiquitous. You can find mushroom corals all over the world. And so they're not necessarily as slow growing as some of the other corals we're hoping to find. And so they're more weedy, if that makes sense. And sponges also tend to be a little bit more weedy in that sense, where they colonize first. They colonize pretty quickly, and they establish themselves more easily than some of these species of corals. And so we're making our way up the slope, hoping to find consolidated rock, which would provide stable, consistent
keep my hand on it so I'm
for
actually
some of the
doing
organisms
something.
we're looking for.
You did say 1, 2, 3, right?
1, 2, 0, yeah.
1, 2, 0.
Yeah.
What is the species of fish that has, it's like a, I think it's called Pacific
More
flathead?
deer. Some of these are pretty darn large.
Yeah. Did we also see a couple of
crinoids, which are those feather stars? We're also seeing other more traditional looking starfish here on the bottom. And actually that dead sponge to the right, middle right of the screen, that looks like a dead heterocone. So now we're seeing a couple different species of sponges. I had thought I had seen a dead heterocone before, so that confirms it. They have multiple different distinct body forms or morphologies. We're also seeing sea cucumbers here, which are related to starfish. They're in the same phylum, the Echinoderm or Echinodermata.
Hey, Lizzie, can you hear me?
Lizzie, can you hear us at all?
Lizzie has left the building.
I just came back because I realized I was talking for a long time and I wasn't getting any responses, and I might have dropped off the link.
Well, we were
true.
We were hearing you.
Yeah, no, it was all good.
Yes, we were hearing it was good, but can you hear me?
I can now. I couldn't hear you before. I just exited VLink and then came back in.
Nice.
Okay, good.
So I apologize if I was just droning on commentary as you're like, Lizzie?
That was all good.
No.
All good stuff.
It was very good stuff.
She's a zombie. She can't hear us.
No, if it was zombie talk, it was very good. So,
Thanks.
no, I was trying to figure out, isn't there another fish that has sort of these spikes that come has a long spike, dorsal spike, and then two pectoral spikes. What is that one called? Something like a flathead. Have we been seeing those? Lizzie?
Oh gosh. I'm trying to think.
yeah.
I feel like I can visualize the fish you're describing, but I'm just also not a fish person, so I don't know if I'd be able to even take an educated guess.
I'm looking through the ID book here. Oh, Pacific flat nose. That's what they're called.
Oh, okay.
Yeah. So we've seen a couple of those too.
Oh, great.
So they have more of a caudal fin compared to the grenadier. That's how I should be able to distinguish between them.
And sorry, can you say the name of the fish again? I'm gonna look for a picture of it. Flat nose?
Pacific flat nose.
Oh, you're right. It does have a caudal fin versus the grenadier just
Yeah.
kind of goes straight back into a point.
Yeah. Yeah. And we heard on earlier watch that the reason they were called rat tails was because the scales would slough off in the nets and then on the tail, and they would really look like a very thin tail of a rat.
Oh, good. That makes sense.
And yeah, and there is one of them.
Big sponge coming
Ooh,
up.
a big sponge.
This is promising.
It looks like another Pharea.
Pharea.
And Guy Cochran in the science chat, as I was chattering away, he said the amount of rubble is actually hard to explain in the normal high bedrock talus model, which I'm not entirely sure what a talus model is, but I'm assuming he's saying based on what they know about geologic formations of bedrock, it's hard to explain how much rubble exists with the potential amount of consolidated bedrock they're expecting to see when we get to the top.
So I'm going to just ask Guy, would you expect a talus rubble that is much finer than what we're seeing here? Is that what is so
I think, is that a coral?
amazing? Yeah, I
Might
think
be a
so.
black coral there. Yeah.
Yeah.
I don't know. It's hard to say. It's like tubular versus fuzzy.
And there's another stalked coral. Yeah.
I don't know if that was a black coral. I think that's something different.
Oh, okay.
That's very exciting though.
Yeah.
Darn this transect. Just kidding. I'm sure if there's one, there'll be more.
We're about 125 meters in, so.
I look forward to the next
Okay.
sighting.
So let me get this straight. That geologist is saying there's a lot of rocks, but it's hard to explain why there's a lot of rocks?
Yeah, so he's saying if you're in the mountains, it tends to be I'm assuming the rubble tends to be close to the mountain. However, this rubble is very low and very wide. So as we're approaching the mountain or the slope peak, the rubble extends much further than he would expect, is my interpretation.
Then there was that giant plain we crossed with no rubble. Is that like a slide of silt came down and covered the rubble field maybe? Or?
Right, it wasn't a very extreme slope, but it kind of looked like a slide area. Like a channel where,
This is one of those occasions where, and it's happened to me before, where I just wish that I understood geology more as a biologist, you know, because everything that we're seeing, the animals, they're living on the geology. And so if I just understood it more. And I guess Guy is saying that the amount of rubble that we've seen for as long as we've seen it, that it has traveled this far. And that the sizes of this rubble to move that far, that requires a lot of energy.
Mm-hmm.
Yeah.
How much of it could have been weathered in place rather than rolled down slope?
Excellent question.
I do not know. So I'm watching the science chat to see if Guy can answer that.
I'm just curious, given that formation we saw earlier where it seemed like there was a bigger chunk that was kind of fractioned. But we'll know as we keep climbing, right?
Yeah. So if it was in place, he would say it would look connected if it was in place.
Mm.
So this is not connected.
Mm-hmm.
Yeah, it's a little chaotic and jumbled. Versus every now and then seeing some pieces that look like they're about to crack that haven't.
Yeah.
And these grenadier are huge.
Big one.
Yep.
Do we fish for grenadier? Is that a commercial species? I think we do, but I think we would export it if we did.
I'm going to see if there's a grenadier fishery.
That's a fillet that we're passing on the right that just exited bottom.
So they're saying, although the look of this fish is not as appealing as other fish when whole, the fillets are a gorgeous light pink color. Its meat is mild and slightly sweet with a flaky texture. So yes, we do eat grenadier. There is a fishery. That's Santa Monica
Yeah, they're
Seafood.
big.
So when you all are in San Pedro and you're off quarantine, you can go to Santa Monica and eat some. Yeah, does it sound appealing?
I know, I'm wondering if they name it something else. Like, it's not grenadier on the menu, but would it be deep sea?
I think it's the other way around, as it used to be more commonly referred to as rat tail, so then they renamed it grenadier.
Yeah,
right.
I don't think I think you're absolutely right. They do call it grenadier. They do not call it rat tail at all.
We have a few coming up here, hanging out.
It'll be a catch of the day.
Yeah.
And that looks like another fillet there on the left, but that's a mostly dead fillet.
I don't think I've ever seen a grenadier eat anything. Yeah, it's not common to see them.
No. I think I saw a crab eating a grenadier once.
Yeah.
The remnants of one.
They might be shy.
What do they eat? They just seem to kind of hang around. So.
I wonder if it's a bunch of small stuff or occasionally one big meal.
Well, we're going to look it up.
Excellent.
Oh, there's two species of grenadier here. We have that bigger guy in the back, and then we have the smaller one that's much darker, with the white tip on his dorsal fin. Oh, Lisa, there's one that you were talking about, a flat nose to the left.
Yes.
I saw he had a caudal fin.
Yes, otherwise,
Nice.
if you just see a shadow.
Very dark.
Hold on.
We are just
Grenadier
about
prey.
towards the end of our 200-meter transect. I can zoom out a bit here on high pack for you on shore, and also Guy. We're looking to see where we're at, if we have any direction, other than continuing on 120 and then altering a bit to get as we go up slope. Keep doing the upslope maneuver.
Yes. Yes, I think.
What is
let's
this
just
coming into
continue on
view?
that.
Is that a Pathy? Or what was that?
It looks like bathy pathies.
Oh, nice. Okay, so black coral. Awesome. We're definitely seeing more and more alive and dead of what
Yeah.
we're hoping and expecting to see.
Yeah, so let's just try and keep on going on this course,
Sure.
and we'll get to the top eventually.
Okay, yeah, we'll keep going upslope. Roger that.
Thank you.
So Taylor, and we can mark this as the end of transect.
Great, sounds good.
Oh, okay. Look at that.
I forgot, what was this, 4?
Yeah. Yep. 4.
Okay.
That's a big
It was
one.
4.
Another huge grenadier.
Yeah.
That would make a
Wow.
huge fillet. Yeah, there are many different species. I think one of the scientists that we work with, Erica Burton, she studied them for her master's thesis. And she said for some of them you can't ID which species they are until you get them on a, out of the water in your hands, and they're difficult to ID, just as we're doing right now in the water on video.
Looks like another can in the water.
Yep.
Some kind of human debris.
Yeah.
I was invited to sail with Mbari on a cruise that came down to just the south side of Santa Cruz Island into the Santa Cruz Basin, because they were trying to ground truth an old dumping ground. It was for military munitions. And so they had mapped previously, found a bunch of targets, and then the goal was to bring the Doc Ricketts ROV back and explore those targets to see how much of it was military munitions versus other kind of junk. And we saw a lot of plastic buckets, big blue barrels, like big metal barrels filled with, I'm not sure what. And we did see a pallet of Fancy Feast cat food as well.
Oh my.
We saw a lot of stuff. It was very interesting. Oh, I'm seeing another species of sponge, that white plate-like sponge. I think they call it Poe, but I'm not sure what the scientific name is. Slowly and steadily, our species diversity counts are increasing. Yeah, so that sponge, that there, is
Yeah.
a dead heterocone sponge, which is a glass sponge. And 2 mushroom corals there just went past the lasers.
Yep. Very good IDing. Lizzie, I'm glad you're here to narrate that part.
There's a few that are pretty easy for me to see or to tell now because I've seen them so many times on the short amount of time that we've been doing Nautilus dives. We've got mushroom coral, we've got the heterocone sponges, Phorea. But there's a lot of other stuff out there that I'm unsure of. Which is why
So
we're
it looks
here.
like we're going.
Which is
And these sticks are either predatory sponges or a type of coral. I'm always surprised to see how similar they are just in a quick glance.
Oh, the white
Those
sticks?
sticks.
Yeah.
Yeah.
Yeah, those are hard to tell from far away.
Yep. Is that a pineapple siphonophore?
I think
We're
so.
going
I
past?
think it is detached and floating away. To me, they look like little hot air balloons because they're tentacles that stick them to the bottom of the seafloor.
You can see some big sponges coming up in the
Yeah.
Argus view.
Hooty hooty hoot.
The anticipation. Ooh, yeah,
Yeah,
another
let's-
large.
Can we? Let's. Oh, that's another Phorea.
Bridge now.
Mm-hmm. Looks like part of it's dying.
One more step,
Yeah.
100 meters, 120.
But we can just keep on going. We don't have to.
Yeah, we can always stop and
Look,
look at
we're-
stuff on the way.
Yeah. We're holding out
Lots
for
of crinoids.
the mountaintop.
Feels like I'm in a spinning class. Just keep going. We just need to get to the
Yeah.
top. That's where the
Yeah.
sunset is.
Don't give up. You're worth it.
What is that?
What was that? The white thing?
Yeah.
That was I think it was a sea star kind of sticking up funny eating or something.
Yeah, it was a sea star trying to impersonate an octopus. That's
Yeah.
what it looked like.
Looked like a little white octopus.
Lots of grenadier here. Another dead sponge. Did that look like a hagfish sort of hiding underneath that dead sponge?
Yeah, I was
I
wondering.
think that,
It was hard to tell.
Yeah, from initially I thought hagfish, and then when we got closer, it kind of like wiggled its tail, and I couldn't really tell.
yeah, it was. But that, if that was a hagfish, that's the first one I've seen on this dive.
It's so interesting that all those heterocones are dead.
Hmm.
Yeah, I was just gonna
But
say
the
we
Phorea
haven't seen
seem
one.
to be doing good.
Seen a live one yet.
Yeah, not a single live one. There's another dead one to the right.
Yeah.
Yeah.
Wonder if this was a good habitat for them, and
Yeah.
no, it's not.
And a lot of this Phorea is either dead or even the live ones have spots
Yeah.
that are not so happy. Oh, there's one of those thicker sticks.
Is that lepidisis potentially? I don't know if that's seen at this depth or not.
Is that bamboo?
I believe so.
It might be.
Let me see. Let me look at a guide. Find a guide really.
I think they are deeper. So yeah, I wouldn't be surprised if it was one because it's rare that we see them in the Channel Islands because we don't have a ton of habitat that gets this deep.
Mm-hmm.
And so I'm that point has stuck out to me. I'm envious of the sanctuaries that have super cool deep sea coral habitat that's deeper than 1,000 meters.
Yeah, but you have super cool shallow habitat, and you know, I think Channel Islands is great. Very cool sanctuary.
Yes, yes, it's definitely cool. We have a lot of more shallow, relatively speaking, for deep sea coral, shallow water sea fans. A lot of the gorgonians that are super high densities, right around the 75 to 200 meter range. And from some of our partners that have been doing research within the Channel Islands, says the future is the way of the gorgonian. So they're not a hard coral. They don't deposit a calcium carbonate skeleton. They have like gorgonin proteins in them versus calcium. And with ocean acidification and the Channel Islands, with a lot of upwelling, because it's at a prominent feature where with the wind direction and the currents, there's strong and periodic upwelling, which mimics a more acidic environment that we could experience due to climate change. And with these really acidic periods from upwelling, it makes it almost impossible for hard skeleton corals like Lophelia or the cup corals to grow very big or form reefs. So as soon as they die and their protective tissue is gone because tissue covers the skeleton, that skeleton is directly exposed to the acidic environment and it just crumbles apart. So normally reef-forming corals, after they die, they maintain their structure, and so other things can settle on them and grow. So like this dead heterocone sponge, for example, the glass silica within them, the spicules fuse, and it becomes a hard surface for other sponges and corals to attach to and grow on, so that's considered a reef-forming species. And Lophelia in other environments is also typically considered a reef-forming species, but in the really variable and sometimes often acidic environment in the Channel Islands, really precludes it from becoming.
Yeah, good.
Yeah.
Looks like the same as, is that?
Yeah.
What is that?
Interesting. Is it dead? Is it covered in stuff?
Yeah.
I feel like,
I think it might be dead something. That's
It looked like a
given
smaller
the odd
version of
color.
the thing we were.
I agree.
Yeah.
We're getting to the mountaintop. So how much further is it in distance
To
for
what?
us to travel? To that little pad
This here?
further up south if we go.
Oh, like the peak?
Yeah.
This over here?
Yeah, exactly.
That is 1.2 kilometers.
Oh boy.
Which I can tell you will take 3 hours or so.
Can we zoom
2.5 hours?
coral?
Sure,
Or the
what was that?
next one we see?
Zoom on which one?
Yeah, we might come across another if you just want to wait till it comes into view, because I'm seeing several now. But when we
Which
come
one?
across one more directly.
Yeah.
the white coral that we're seeing?
Yeah. There will be more.
How does that timing match up? That's just a rough calculation. It'll be about 2.5 hours if we continue steady around this speed.
Oh, here's some more coral. This one's branching. I think they're bamboo corals.
Yeah, let's just check out the bamboo coral.
Yeah, to the right, could we zoom, and then the folks analyzing this later can get hopefully a more